this is going to be a very informal bioinformatics talk. And the most important thing I want to get across today is not to teach you how to do bioinformatics, but give you a take, because you can't do it in an hour. You just can't. What I want to do is kind of show you what's out there. And I want to show you how this can completely change the way you approach your research. To do this, though, in the amount of time I'm giving, I'm using the top-of-the-line commercial software platforms, NextBio and IPA. Okay? Put this up here. Now, they might seem, there's kind of a mentality that, people don't think they should have to pay for their bioinformatics, or that it should be offered free. But, we can all make our own antibodies if you want to do your own Western. Most of us don't. It'd be cheaper that way, though. That I would rather spend my time, instead of focusing on the tool, focusing on what the tool can do for me. And that's why I use NextBio and IPA. Just to give you a little kind of background into my, how I came up. I started in graduate school. This was in the year 2000. It started, basically the arrays kind of took off during that time. And I was at the bench. I didn't have any computer skills or not an extensive mathematical background. But I quickly realized that it's a lot better to look at all genes rather than just one gene. That the ability to look at the entire genome at the same time is incredibly powerful. And that's kind of why I got into bioinformatics without any computer skills. Eventually I thought, well, I'll just get into it. I'll use these commercial software programs and pick up the freeware, maybe learn some programming, but what happened is the freeware didn't catch up to the commercial software. In bioinformatics, you really do get what you pay for. And that with a lot of these bioinformatics programs, what you're paying for is not the tool itself, but the database it's tied to. And so I'm going to explain some of these databases in NextBio and IPA and why it's so important, or why you have to pay for this. So in order to kind of show off what the software does, and give you an idea of what bioinformatics can do for your research, I'm going to use an example from a high school class that I've been teaching for the last 2 years. And these kids get it. So what I'm going to do is basically take you, using these two programs, we're basically going to analyze the data set that I had my high school class do. What that means is if they can do it, you can do it. You totally can do it. And in fact, it would be better if you do it. It's always amazed me how somebody would take, they spend all this time and money putting this this research project together, spend all this money, and then at the very end when, the cool part, when they get the results, they give it to somebody, say a computer specialist or a bioinformatician or a biostatistics person that doesn't really understand the biology. You all know your biology. You should all be able to look at your own research, because you're going to have the insights to find, whatever potential, valuable information that's going to come out of it. The computer science person is not going to understand. This software, the commercial software right now, will allow you to do that. So why is this important? I'm going to tell you right now that we could all stop killing mice and growing cells, and, no other scientific experiments could stop today, and I'd have enough data for several careers. There is so much data out there, and this is one of the more popular repositories for that, Gene Expression Omnibus. Has anybody heard of GEO?
Yeah. GEO, yeah.
GEO, yeah. If you get funded by the federal government to do research, you have to make your data available. I want to show you something here, and I took a screenshot today. If you look at this series, this is the amount of experiments that are in GEO right now. It's almost 70,000 experiments containing almost 2 million samples. And these are all kinds, this is petabytes worth of information. What happens to me a lot of times, and I guess what I'm trying to get across is that there is so much data out there that the real true power of bioinformatics isn't just to analyze your own data, but to take your data and compare it to all the other data that's out there. Here's what usually happens is when somebody will come see me, they want me to do support on their grant. And they say, hey, Mike, can you do a power analysis? I'm going to propose, sequencing 10, lung tumors. And, we're going to do this, but it's a special kind of form of lung tumor and, but this is what I'm going to propose. And the first thing I ask them is, have you checked to see if anybody else has done that? If there's any other samples out there? And usually the answer is no. So I'll just put in a keyword here in geo, and sure enough, usually I find not only exactly what they wanted to do, but it has more samples and better controlled. This can save you a lot of time, that you don't have to do those preliminary experiments. This will do your preliminary work for you. Okay. So to demonstrate this, like I said, I'm going to basically go through an example I did with my high school class this last year. This is at Green Valley Ranch. If you're driving to the airport and you get off of Pena Boulevard, it's that first exit to the right. This is this isn't a special school. I believe 60% of the students get free lunch. There's a 95% are minorities. What I've been doing is teaching a senior class big data analysis, and at the very end we do bioinformatics. And the reason I'm able to do bioinformatics with these kids is basically we have this commercial software that I'm going to demonstrate to you. And it was a diverse group too. This isn't your AP students, or definitely not all of them. 3 of my students, it was a question whether we're going to graduate or not from high school. And one kid who will probably be the first one in his family to graduate high school is just an absolute genius. So there's a huge span of skill level there. Oh, so I asked one of the students, I said, what did you guys want to do? And, I said, all this data's out there. I gave him told them, think of any disease, anything you want, we could probably investigate. One of my students, as I said that, said, well, I had an aunt that just died of breast cancer about 2 weeks ago. I go, well, let's look at breast cancer. And so that's exactly what we did. So I basically talked about breast cancer, gave a little background on how this happens, and some of the forces that cause this. So this is basically giving them targets to look at as we analyze these data sets. Obviously you have oncogenes. You can consider these as kind of the criminals of cancer, right? These are things like, and tumor suppressors, which are, like the police department in your cell. That every biological condition is kind of a balance between two forces, one going one way and one going the other. And it's that balance that determines, what your biological system is going to do. And again, oncogenes, growth factors, signaling molecules, especially cell cycle genes, cell progression. Anybody here work on cancer? No? All right, cool. Not breast cancer, right? No. Okay, I was going to say you get some free information today. Telomeres, blood vessels, recruiting blood vessels, and then tumor suppressors. And these would be things that normally inhibit or control these oncogenes. Here's another important thing to understand about bioinformatics, is that if you're looking at a disease and you're looking at gene expression, if everything worked correctly, you wouldn't have the disease. So one thing to think about as you look through your gene list, if it is a disease, if it's something wrong with the organism, you're probably going to get altered signaling pathways. So I wanted to basically explain that to him. And we talked about, obviously breast cancer just doesn't appear. It's a process, and there's defined stages of breast cancer. And as we were looking through this, we noticed that there's this huge drop-off in survival rate at stage 3 breast cancer. So we thought maybe stage 3 would be a very good developmental stage of this cancer to look at. Not only that, but whenever you're asking when you're looking at all the genes, it's very good to ask a specific question. If I just wanted to find the genes that are different in breast cancer to normal tissue, I'm going to get hundreds of thousands of genes that are different. It's going to look like things blew up. So what's nice is I can compare two similar conditions. So basically we wanted to investigate stage 3 and compare it to the 2 stages below that. So in effect, what I'm doing is I'm comparing cancer to itself. It's just a different stage. That if I compared cancer to, say, normal tissue, I would get lots and lots of stuff different. This technology, especially when you're doing gene expression, works better when there's only slight differences, when these systems aren't completely different. And I excite them in this way is that, listen, there's a lot we don't know about breast cancer. So the things that they see, you might be the first person to even know this about breast cancer. And this happens to me all the time, that even if somebody publishes on a data set, I find they only take like a small sliver of the information that's contained in all that information. If you go back, normally I can get 20, 30 times as much information as what they've actually published. So just because somebody's published on a data set doesn't mean there's not diamonds in there. Okay. So let's get to the actual, so what I'm going to do is basically kind of go through what I did with this class and what I had them do to analyze breast cancer, to come up with their own hypothesis of what's going on.